1, A. Question 2, B. Question 3, B. I hope you have enjoyed the second round of the Chinese challenge. Next, we have Team 1 from Westwood Secondary School to present their approach to the problems. Let us welcome them.
Good afternoon, everyone. We are from Westwood Secondary School, and we will be performing a skit and host a Q&A session after. The skit will be about an episode of Crime Watch. Without further ado, let us begin.
Welcome to today's episode of Crime Watch. I'm your Earn and Miss Candy. We are your hosts for today. Two men are trapped at Singapore Customs. Let's watch this video to find out more. Scientists to explain more.
I have finally gotten the results after some tests. The first test I did is to differentiate the real tiger bones and teeth from the fake ones. This is done using UV fluorescence. If the tiger bones and teeth are real, it should turn pale blue. And it did. Therefore, I concluded that the tiger bones and teeth are real. The second test, DNA analysis, is used to tell which species the tiger bones and teeth belongs to. The extracted DNA was amplified by polymerase chain reaction. Primers were used to select specific regions for amplification. Restriction enzymes are then used to cut the DNA into specific length. The DNA would then move through an agarose matrix. The shorter strands will move through the agarose matrix faster than the longer one. The DNA strands is now separated into bands according to their length. The patterns of bands can be compared against the database to determine which species the tiger bones and teeth belongs to.
Let's get Dr. Sam to discuss more on DNA extraction.
I found human blood on the packaging. We can check if the
blood samples I collected is too small for the test results to be conclusive.
No worries, with nanotechnology, it can be done. Part of the DNA sequence is covalently attached to the glass surface as well as gold nanoparticles.
Why use gold nanoparticles?
Gold nanoparticles have a larger absorption cross-section compared to other organic substances, allowing easy detection by the UV spectrometer.
What does absorption cross-section means with respect to nanoparticles?
The absorption cross-section is a measure of the probability of the absorption process. If a substance has a strong molar absorptivity, which is how strong a chemical species is able to absorb light, then the absorption process will be greater.
Since we are using nanotechnology, does it mean the smaller the better?
That's not necessarily true. Comparing a 10 nanometer and a 20 nanometer nanoparticle, a 10 nanometer nanoparticle will have a smaller absorption cross-section as the amount of atoms is lesser and there is a smaller area involved, thus the amount of absorbance is lesser compared to a 20 nanometer nanoparticle.
Nanotechnology sure have many advantages.
It does, but it's so small that it's not like a body. It's able to pass through the cell membrane of our cells and interact with the biological system inside and cause problems to arise. There is also another problem involving the use of low nanoparticles to analyze DNA. The low nanoparticles tend to precipitate out of the aqueous mixture.
Can you do something to stop that from happening?
Yes, we can. We have to use a chemical stabilizer in the form of a surfactant coat. There are three structurally similar commercial stabilizers that we can use. When it's coated on the surface, you will use alkyl, hydroxyl, or carboxylic and glues your identical alkyl chain length, which I chose the one using alkyl group. As we're using water to wash off unbound gold particles, an appropriate surfactant coat must not be soluble in water. The alkyl chemical stabilizer will be the most suitable as it does not form hydrogen bonds with water. For the hydroxyl and carboxyl stabilizers, under the exchange with the alkyl group, their solubility will be very high, which means we could use information should the bound gold particles be washed away.
If we want one gold nanoparticle to collect 1% of DNA, what is the minimum concentration of DNA that we need?
Assuming that the detection limit of a UV spectrometer is 0.01 absorbance unit, and the absorption cross-section of a 40 nanometer gold nanoparticle is approximately 3 times 10 to the power of -11 centimeter square, the minimum concentration in moles per liter that can be detected in the cuvette of 1 cm half length will be equal to 1.27 picomoles per liter. This is all I have for now, but I'll be back again if I find more information.
Now I understand more about DNA extraction, but I don't understand why the police did not use X-ray to scan the luggage. It seems that the tourists just in the passenger gates and something will sensor tigers in their bags.
I guess that they are using nanotechnology.
What is nanotechnology?
Nanotechnology is a manipulation of matter on an atomic or molecular scale.
How is nanotechnology used in airport security?
It is used to detect homemade bombs and illegal items. Airport security systems such as the nanotechnology scanners is able to detect explosives and chemicals. The system works by using infrared laser to scan the chemical fingerprints found on people's clothing or on their baggages. From this, the scanner is able to identify any residual molecules that are linked to explosives. All of this is because of carbon-based nanomaterials which are graphene, fullerenes, and carbon nanotubes.
What are carbon-based nanomaterials?
Graphene is a single atomic layer of graphite arranged in a honeycomb crystal lattice. Let me show you an example of how it looks like.
It looks interesting, so that's graphene. There was a fullerene.
A fullerene is just an enclosed structure, usually in the shape of a sphere. Take a look at this football. This is the shape of a fullerene. The corners of the football are the carbon atoms. There are carbon atoms along the lines which are the bonds between the carbon atoms.
Hey, the football is a mixture of pentagons and hexagons. How many pentagons are there?
For football, there are 12 according to Euler's formula.
Only 12? I thought there are more.
Look again, only 12 are pentagons, the rest are hexagons. The pentagons do not touch each other. The black ones are pentagons and the white ones are hexagons.
How many hexagons will there be?
20 hexagons.
Is it always 20 hexagons and 12 pentagons?
Not really, only when there are 60 carbon atoms.
What happens when there is a different number of carbon atoms?
This is what you get. Not the perfect round shape like a football, there are only 36 carbon atoms here.
Interesting shape. There was another tube.
Take a sheet of graphene and roll it up. Roll it from edge to edge, corner to corner, or side by side. you'll get different patterns or different properties.
What do you mean by different properties?
Sometimes, depending on how you roll the graphene up, it can become metallic or non-metallic. It can conduct electricity or become semiconductor, which means it is usually an insulator, but when it's given extra energy, it can become a conductor and conduct electricity. Hence, this property can be useful as a sensor.
Now I understand carbon-based nanomaterials much better. We have come to an end of Crime Watch this season. See you again next time.
We're now come to the end of our skit and we'll open the Q&A session now.
Thank you. So a quick question regarding the kind of surfactant one should be using under this kind of competition. And what you guys seem to suggest is that alcohol-coated surfactant would be more appropriate. So what do you think it, why don't water-soluble surfactant be advantageous? Because ultimately you want to be able to measure that you do this, right? How are you going to suspend it in water if they're insoluble? So my question is, you seem to suggest that the alcohol-coated surfactant would be more advantageous in your detection scheme. I want to play the devil's advocate and say it's no. Okay, so why do you think it would be better to have a aqueous soluble surfactant instead? Think of it this way: if you have your gold nanoparticles that are all with this alcohol coat, right? You get them. You kind of suspend in water, right? That's because it makes it so hydrophobic that it just crashes out. Isn't that a problem? So am I correct or are you correct? So I'll give you a chance. If you were to, what would be better? If I tell you now, the answer should be the C, which is a surfactant that will allow it to suspend in water. Could you then explain, why would this be a much better system? Yeah. So if you have a carboxylic acid group, why is this more advantageous? And why would this surfactant allow the gold nanoparticles to be more to suspend, to dissolve in water? No? Good. You have an answer? If not, then we just move on. Sorry.
Hi. I have two questions. The first one is back to the first slide. What is it when you put the a real tiger tooth under the UV fluorescence? Why is it that it will move?
Since the synthetic bones and teeth have different chemical composition from real one, it should react to the UV fluorescence differently because different chemical composition will absorb and release the different wavelength of ultraviolet differently.
So why is it that the tiger bone will make it blue?
The tiger bones and teeth reflected is actually longer than ultraviolet. So it will like drop down to the visible light range. So you can actually see.
Okay. The second question would be, you mentioned that in the soccer ball, the pentagons are not touching each other. Is there a reason why?
Basically, the hexagon has 3 vertices which pentagons are connected to. So each hexagon, the method of a hexagon and a pentagon connect to is by 3 vertices. As seen in the football, in hexagon there will be a middle will be a pentagon and the top vertice, and the two other side vertices will be the 2 hexagons. So using this example, we can say that the pentagon and hexagon do not touch each other.
Further to this question, because you said you use UV fluorescence, so you are heating the phones with UV light. When you use, correct?
Excuse me, can you repeat?
You are using UV fluorescence, so you are heating the phone with UV light itself. So when you say it is red, it will turn pale blue. When your pale blue means it's just pale blue, or is it fluorescence? Or is it there's light actually coming out? There's actually this bright pale blue light coming out.
Yeah, actually there's this pale thing.
So there's actually a bright pale blue thing that's coming out.
Yeah.
No, because I'm just curious, because I work with UV lights on crab. I work with crabs, so I'm very crab. Okay? So I use UV light and I hit on the surface of the crab, that means the crab shell, and I see what kind of reaction that it does come up with. So when you talk about pale blue light, I don't know whether, because if the crab does not exhibit any fluorescence, the carapace does not exhibit fluorescence, it's just a pale blue color, like a normal. But if there is fluorescence, then there is this very intense bright light that's being shown. So I just want to know when you talk about pale blue color, exactly what kind of frictions you're talking about. No worries. In your video, you didn't show anything about how do you, because in your video you show the person you caught and then open up the bags. Okay, I have to tell you this, in real life, the custom officer will not open the bags themselves. They will make you open the bags and take out everything yourself. And then when they see something, they will say, why? Like, okay, but that's one thing, that's the side. It's not that I got caught, like, because I see people doing it. Okay, then the other thing is, how do you know those, is there any way? Okay, I saw you were very good, you were actually unwrapping it with newspaper, then the leaf, then plastic bag, then newspaper, then the leaf. If let's say is there any other way we can actually tell that these bones are actually from India and not from Bali?
We can actually use ISO signatures to tell where does this bones and teeth come from.
Is there a simpler way? Because as a custom officer at Changi Airport, you don't have so much time to do this ISO testing. Okay, let's say you have to make a decision within the next 5 minutes to whether to detain this couple or not. What do you think you can check?
Is it possible that I actually like check the travelers commuting records for that?
Check the passport.
Yeah.
Where the boarding passes and everything.
Yeah.
Right? Okay. Anything else? The most important thing, anything else that you can actually see from the packaging itself? Is there anything you can actually see from packaging that says that as a valid reason to detain the couple, so they can do all your further tests?
Can you hang on for a while? So the question is to tell which country the tiger was came from.
Okay, you see a packet of bones. First, how do you know that they are bones? You need to do a lot of tests, right?
You can check for blood stains found on the bone.
Yeah, you can, but you can also, but what if the bones are actually older? That means there's no more blood stains. What is the first thing that you can actually check to say that these are animal bones which require further testing, so that you actually have reason to detain the bone? Because, for example, like you need time, you cannot bring a UV light there and shine on the bone, right? I mean, you cannot, there are portable ones. Okay, but you need to have your DNA test and your ISO test and all this. So you must have a valid reason to say that I need to detain you on the basis that you are probably bringing in materials like this. Simple. Look for the surface on the bones. On the surface of the bones, you see, under, if you have a magnifying glass, you can put your answer. Okay, all you come in.
So you're saying that since the bones are not really solid and like hard.
Yes. Yeah. Thank you.
We will release the results for the second judges challenge. And the winner goes to Matthew from Hwa Chong Institution. Please come down to receive the And the second winner is Go Big Night from Chongqing Island.
Hey, what is this? Is this smuggled bones in your bag that I see?
No, this is actually imported from Bali. They are plastic mixed with plaster in order to simulate the look of real bone. I bought it as a souvenir.
Something isn't right here. Please wait here with our security guard while we take this for examination. Excuse me, could you help us to check if these bones are real or fake?
Okay, let's see.
Okay, let's see this bone. There's other uneven surfaces. And
What are these black dots over
yeah, and
here?
there are black dots all over the bone. However, we cannot confirm that these are an animal origin just because of these two factors. We just need to do other tests.
What test should I be?
How about the X-ray spectrometer? And the main difference between a real bone and a plastic bone is due to the presence of calcium. Calcium is found in our daily dietary products and vegetables. And so, to operate a X-ray spectroscopy machine, the first thing that we do is to shine a beam of high energy electrons on the surface of the bone. And then, an electron from the inner shell will then drop.
So what happens to this hole?
For this hole, an electron from the outer shell will then drop back into the inner shell to replace the calcium.
Oh,
And through this, energy is released from the bone. And to measure the light, use an energy
So are these bones real or fake? Can you just come? Why? Okay, thank you. I shall not confront the suspect. Joe, what happened? Why are you bleeding so much? You're dead. What? Hello, is this the police? Yes, someone has been murdered. Yeah, this is the airport. Yeah, come here. Oh, sorry, 3. Yeah, can you please come over? Ambulance? Yeah, okay, come. No, airport, terminal 2, 3. Yeah, please come over now. Someone has been killed. Yeah, faster. Yeah, there is a...
Okay, let's examine the subject. Seeing as most of the blood has come from the security officer, DNA from the culprit must be in low concentrations. However, this is not to worry. We can utilize the recent technological advancement of gold nanoparticles. By introducing gold nanoparticles and a solid glass surface with the potential suspect DNA, we can use a DNA to DNA hybridization to determine if they are matched, and hence we can deduce the culprit. However, the following drawing will occur. Firstly, the gold nanoparticle with the suspect's DNA attached will form hydrogen bonds with another gold nanoparticle. The gold nanoparticle with the suspect's DNA attached will be fully bonded to the DNA on the solid glass surface without the unknown DNA being a bridge. The gold nanoparticle with the suspect's DNA will bond to an unknown DNA, however, the remaining unbounded segment of the unknown DNA will be bonded to another gold nanoparticle. Lastly, the gold nanoparticle is in...
I'm sorry, but I don't quite understand what you're saying. Can you simplify things a little?
Okay, basically, if the DNAs do not bond in a particular order or way, which is the DNA of the gold nanoparticle is linked via the bridge, which is the unknown DNA, to the DNA on the solid glass surface, the result will not be achieved, and hence the effectiveness of the test is reduced. Furthermore, this test is not really confirmatory. It only determines the similarity level between the two DNAs, which is the unknown DNA at the crime scene and the suspect's DNA being tested. If there are people with similar DNA, for example, siblings, it could result in a false positive.
But I did hear that the suspect has no siblings, so here is not the
Simplifies things very much.
Okay. So now that we have determined the identity of the suspect, we have to find out methods to improve our airport security. How do you suggest we do so?
Aha, I have the solution. The recent advancement in fooleries may help.
Oh, what are fooleries? What are those?
Okay, so we all know of an element called carbon. So carbon atoms can be arranged in different manners to give different forms. These are called fooleries. There are 3 main types of fooleries: carbon nanotubes, monkey balls, and graphene.
Oh wait, so with this small micro objects, how does it help? I mean, we need shields, armors, what are you talking about?
Oh, these are not micro objects. They are even smaller, or what you call nanomaterials. These small little things can be used for very different purposes. Let me first explain how they work. In general, they have a high melting point which we can exploit. A buckyball is actually a huge space that can enclose atoms of small molecules that we can use. Yeah. So, and carbon nanotubes are double bonded, which means that one carbon atom shares 2 electrons with one another. As such, their bond is stronger and thus they have superior strength. Yeah, properties can also be adjusted by the chiral vector. The shape and the arrangement of the carbon atoms affects its electrical conductivity. Graphene also has vast moving properties.
Sorry sir, but my knowledge of science is at the O level, so I don't quite understand what you're delivering. Can you simplify things a bit?
Okay, I'll simplify. Okay, so buckyball is just a huge space that can enclose the needed molecule or atom for what you need, so you can use it. Yeah. For carbon nanotubes, with their high thermal conductivity and high melting point, you can use it to heat and accelerate electrons in the X-rays you use, hence increasing the energy efficiency.
So these things can detect things better.
Yeah, that's true. Also, the buckyballs and carbon nanotubes can work together to improve the strength of your armor right now.
You mean bulletproof vest?
Yes, bulletproof vest.
Okay.
Yeah.
So how else can you use these carbon nanotubes to prevent such accidents from happening again?
Okay, so I mentioned that it can be used to increase the energy efficiency of the airport scanners. It can also detect more stuff and will allow more things to be detected. And it can also be used to enclose bomb blasts to reduce the area of the bomb blast.
Okay, thank you very much. We shall take your considerations to improve our airport security.
2 years later.
Wait, what's this? Bones? Bones again? You, come here!
Oh, what? Is it working?
Come! Nanotechnology for better future. Thank you.
Thank you. We will now have a Q&A session on Team 1 from Hwa Chong Institution.
Okay. If you are a very good smuggler, do you think it is actually an efficient way to smuggle your tiger bones together with other things that can avoid detection by the X-ray machine?
It's sometimes called like hiding the truth in an obvious spot. So the more obvious it seems, the less likely it seems that there is tiger bones.
For example,
It's using psychology. So sometimes you hide stuff. Sometimes you let your things remain the most open spot to make them like question why is it so open, and sometimes they may even miss the open spot.
like wear a tiger tooth on your neck, right? You walk right through the custom, is it? Very good psychology. I better warn the Changi airport of you guys. Actually, I don't know whether it's this. I'm not teaching you how to smuggle things, but I have seen a lot of people who go, say for example, to Hong Kong. They come back with a big box containing their whole stuff. See, and then within the roast up box, I don't know what else you can put inside there in addition to the roast duck. with the tiger bone pattern. Now, we do not know whether this tiger bone is real or fake because usually, you know, especially when the flight is going to take off, you're like probably the last passenger, the airline just make sure that you just go on to the flight and then that's it, so that you don't waste whatever fee or do not pay extra fees whatsoever. So as you said, it's actually sometimes obvious that you just wear the thing and then you just walk right through and then you don't pay. Okay. In theory, in Singapore, you're not supposed to bring any birds into Singapore, cooked or raw. And there's no chicken, no ducks, no everything. But you see people coming back from Hong Kong with a roast duck. Almost every uncle, auntie will have a roast duck. Yeah. So in theory, no. So in fact, I think this is something that I think a lot of smugglers they do. They actually make use of uncles and aunties, alright, or the least suspecting ones to carry things for them. Now, Singapore, we have tiger bones on sale. If you go to certain shops, in certain places, you can actually see them on sale. And we know that based on the age, based on the look of the specimen itself, we know that these are not old bones. These are not bones that are like collected 50, 60 years ago. Okay, these are recent products. Alright, and this is why there's actually a group in Singapore that actually goes around actually looking out for all these bones, and then they will actually make sure that they are being graded and whoever that's responsible to prosecute. Okay?
So, I have a question. In the beginning, you showed this X-ray spectroscopy. My guess is you're talking about energy dispersive X-ray spectroscopy.
Yes.
So, what kind of machine can you find this technique in? What kind of machines have this X-ray, energy dispersive X-ray spectroscopy?
It's probably those machines where you would scan through a certain material to determine its chemical composition through spectral lines. Through observing the atom spectral lines, you can determine the chemical composition of the object, and hence you can find what's without.
I guess I was looking for a specific name. For example, the electron microscope has such techniques. However, so you have a transmission electron microscope, you have a scanning electron microscope. They can incorporate such a technique in there. But my question would be then a follow-up question. My original one would be: can you stick a whole tiger bone into such a machine?
Can't actually. Because actually the tiger bone, actually we can reduce the tiger bone size to fit it inside.
Okay. So between a transmission electron microscope and a scanning electron microscope, which one do you think will be more suitable for you to slice your tiger bone and put in, so that you can do this energy dispersive X-ray spectroscopy? You get 50% chance. It's either TEM or SEM. So you picked the SEM. Are you sure about that?
Not pretty sure.
Okay. Final answer.
Yeah.
I'll bear that in mind.
Thank you for the nice sketch. Just one question regarding, you talked about some problems with the DNA detection system. You talked about some drawbacks. How about some suggestion? How would you try to improve this detection scheme, which is obviously it's deliberately flawed? But how would you, as scientists, forensic scientists, try to improve this detection scheme? way you want to enrich the DNA material? Is there any way you want to improve the low level particles? Thank you.
We will now proceed to the third round of the judges' challenge. Students, please get your handbooks ready again. And please follow the instructions on the screen.
Before we proceed, let us take a look at the answers to these questions. Question 1, C. Question 2, A. Question 3, B.
I hope that you have enjoyed the third round of the judges' challenge. And the winner for the third judges' challenge is Sim Tao Ming from NUS High.
This year marks the 14th year of chaos. The Gifted Education Branch is able to organize this with the support of sponsors, the Defense Science and Technology Agency, and DSO National Laboratories. We have also benefited much from the collaboration with the science faculty, National University of Singapore. We would like to take this opportunity to acknowledge and thank our partners today. May I now invite Dr. Tan Lee Hyong, Deputy Director, Gifted Education Branch, to present tokens of appreciation to the judges. Dr. Tan.
And our judges are from the Department of Biological Sciences, Dr. Ng Yang Kim. From the Department of Chemistry, Assistant Professor Chan Yi Tan. From the Department of Chemistry, Assistant Professor Ang Wee Han. And from the Department of Mathematics, Dr. Lin Shi Han. Thank you, Dr. Tan. The judges will now take their leave to deliberate on the final
going to represent our panel of judges to give the judges feedback on the presentation and the written reports that we received that we graded for this competition. First and foremost, I would like to congratulate you guys for giving a very good submission. It was very encouraging for us to receive this submission. They were all of very, very high quality, and in many cases they were all very insightful. So please give yourself a round of applause for that. Okay, so well, we think that there are still a few points that you guys can work on, to improve the report for your future submission. So the first point we'd like to make is, in some this exercise is meant to be a stimulating exercise for you guys to discover the science involved in each of the question involved. Sometimes there isn't a right or wrong answer. Okay, it's meant to be open-ended in that way, and we want you guys to discover and give your perspective and argue your case. And sometimes the answer that they were given was a bit too short. Bear in mind that it is open-ended, so we'd like you guys to give more, to build your argument a bit better, to build your case a bit better. Avoid giving short answers. Okay. The second point is regarding the source of the information that you guys rely upon. Okay, for the answers. I know it's very convenient to click on Wikipedia, and there you have, the world knowledge at your fingertips. But bear in mind that Wikipedia is not authoritative information. At least it's not in the scientific community. Okay? There are flaws in Wikipedia, okay? And you guys should be aware that when you obtain information from Wikipedia, they may or may not be verified. Okay? What we consider to be correct sources, primary sources of information, your correct primary sources of information should be from textbooks, from journal articles. In fact, that's what we use routinely. We use journal articles. We routinely cite journal articles. Okay? Wikipedia is great. It's convenient. Okay? But you should verify it. And sometimes in your Wikipedia pages, you do see that they cite where they get the information from, right? So you can cross-check information, okay, to make sure that what you obtain is accurate. Okay. The last point is a minor point, but we notice some of your answers are not answering to the point. So just be more cautious when you're answering the question, because even though our questions are open-ended, they are meant to ask a specific scientific question, and just try to be to give relevant answers to that point. Okay. The second part of my feedback will be regarding today's presentation. I must tell you, the judges were all really impressed with the theatrics and the show that you guys have put up. I mean, it's really amazing. So again, it's a job well done. We were particularly impressed by the fact that you guys are able to link up. Before this competition, the judges were asking questions with regards to chemistry, biology, and mathematics. And even though the questions were quite distinct, you guys managed to piece them together, weave them into a really nice story, and we really enjoyed it very much. So well done. I guess the point that we were very impressed with the quality of your presentation. Just one minor point that we would like you guys to take note is, even for us, we give scientific presentation. It is important when you present something, whatever information you put up there, what things you say, you must be able to substantiate. So there's no point putting something that you don't quite understand. Okay? You should, when you put something out there, you're subjected to scrutiny. So you must, when you say something, when you describe a process, you must have some level of understanding. You must try to understand, what that particular thing is all about. okay, so that when we probe you further, you would challenge us, and we can challenge you in an interactive way. But this is how it's being done in the scientific experiment. So that is something that we hope you guys will improve upon. And lastly, the judges would just like to mention to you guys that science. This is what science is about. Science is not always there's usually a right answer, but the process to getting to that answer, is a process of discovery. Okay, there are things that we do not know, and we're trying to discover. And chaos was framed in a way to help you guys realize, to go through the scientific process, and using this methods to try to solve daily challenges, because this is what we guys do on a routine basis. And I think you guys have achieved the objective of chaos, and we are very happy to see that. So again, on behalf of the judges, we thank you for your participation and your job well done. Thank you.
I have 2 sheets of questions for the biology section. Okay, I will answer them. Not that I can give you all the correct answers, but at least this is as far as I know. You see, scientists are not answering machines. We have to do research because there are a lot of things we don't know. Okay, so we are waiting for you guys to come and join our workforce so that we have more answers. Okay, to provide to your children or your grandchildren. Alright, the first biology question. Couldn't one look at their body parts to determine where it came from? Yes, we can. But many of the members are quite smart. They actually throw their body parts away. Then, without their body parts, then in the transit area, then you really do not know where to come from. Alright. Then of course, why also don't the police arrest a suspect then interrogate? Usually what they will do is that they will bring the suspect to a corner and then they will do the start of the interrogation. Now, for the police to actually detain anybody, there must be a valid reason. And this is why I ask some of you, given just a magnifying glass, the most simple equipment available that you have, how do you determine that these are animal bones? At least give them reason that say that, okay, come on, I suspect you are having animal bones. Okay, whether be chicken, duck, or tiger, or bear, or hippopotamus, doesn't matter. Alright, just tell them that these animal bones, they require further testing. I need you will be detained until we are able to confirm the identification of these bones. Otherwise, they sue you for harassment. Alright, and they will lose your job. Alright, so you must give them a valid reason that come on, we suspect these are animal bones, and that's what we need to do on it. Why does the suspect wrap bones in leaves? Well, I don't know. Leaves are used to wrap a lot of things in the past, especially in the rural areas. Southeast Asia is, we still have a lot of rural areas where they still use leaves to wrap things. I don't know, go back and ask your grandmothers. In the past, in Singapore and Malaysia, they wrap chee chong fan with simple eye leaves. You know what's simple eye? Simple eye is a Malay name for a very common plant that is found on the Cambridge and found everywhere. The leaf is this way, alright, and then it produces bright yellow flowers. So this leaf is very broad, so they actually use this to wrap your chee chong fan, your food store, your whatever food. And then they only stop using this when we start to have plastic sheets, then we use plastic sheets on top of newspaper. Then of course now we have styrofoam boxes, so we don't use that kind of things anymore. But if you go to certain countries, you go to some parts of Southeast Asia, you will still see them using this leaf to wrap things. Alright, so you won't be surprised that they actually use the leaves, any leaves, big enough, broad enough, alright, sturdy enough to wrap the bones in. Okay, so does that answer the question? Yes? sensor. So if you actually have a keychain of those prospects, you know, it will come out differently. And that's when they say, oh, can you come out and take out empty your pocket and then go and stand there again for another, 30 seconds? So, depending on which airport you're going through, it is possible that you actually smuggle things out that way. I have a colleague, because in Sri Lanka, you're not supposed to bring any living, non-living tissues out of Sri Lanka. They are very risky. If they're caught, you are dead. But yet, that guy managed to smuggle some of his specimens out for his study. Alright, he just, because his animals are tiny shrimps, as you know, prawns, shrimps. So they put them in lavender tubes. Okay, and he actually had some 20, 30 lavender tubes in both his pockets. He just go right through the machine like this. He did it, then we say, walao. I couldn't dare to do it, boy. Even though I'm auntie now. But no. So it is actually possible. Alright? But you see, bones, tiger bones, alright, are big enough, are so big, not hide in your pocket. If you say you hide a tiger teeth in your pocket, like just what the last group of presenters has said, you know, you can just do it. You do it just so blatantly, right? You think everybody that is a fake, you just wear it around your neck. And nobody will suspect that you are actually smuggling the thing out. But, for heaven's sake, please don't do these things like this, okay? It's totally illegal. Okay, I don't want your name to appear in newspaper. Wow, Singaporean not smuggling tiger bones. Someone from our school. Okay, so please don't do this. Okay, the next question is, could you further explain the pale blue fluorescence on the bones due to UV? Now, the fluorescence itself now, many of the organisms are able to, what we call, because we all know that UV lights are actually harmful for living things. Many of the organisms are able to make use of this UV radiation and convert it to something else, so that the radiation will not penetrate into the organism itself. For example, in corals. Corals, okay, you look at them, yes, they are very pretty, they are colorful, but under UV radiation, some of these corals actually fluoresce, or they glow with a different color. That's because they're able to convert this UV energy into something else, so that this light is being reflected or deflected out of them, from them, so that they will not penetrate into animals and create any harm. Alright, the bones itself, well, we know that different animals have, well, I mean, yes, all the tigers have, all the animals eat have different diets. So many of them actually incorporate whatever minerals they eat. They take it into their bones. So the bones itself, for this, so it's possible that, you know, some of the minerals actually inside the bones will actually react, will actually fluoresce or glow. Alright, when you actually shine UV light. In fact, it's very simple. I was at the water plant, and they actually have this panel to showcase UV light. And it was very interesting because if you go to the toilet and you don't wash your hand properly, you go under the UV light, then it fluoresces, then it glow. Then we all know that you didn't go to the toilet and wash your hand. So it's the same thing. Alright. Now, not all the bones may react to the UV light. Some of the bones will react, the fluorescence will be stronger, some of them will be weaker, depending on the different type of materials that's deposited within the bones itself. Any questions? literally destroyed all her finger, all her 5 prints, both hands, so 10 prints. And then, based on whatever scar that she actually used that as the new print, and then she came to Singapore. And then, unfortunately, the second time when she came to Singapore, her daughter was murdered and wrapped in a box and thrown into some forested area. Alright. And all these things came out because the daughter was murdered. Otherwise, we wouldn't have known that she actually has come into Singapore using another passport. Okay. Alright? Okay, any other questions?
So let me go through the other question that we got on the nanoparticle stuff. So the question is, what other uses can the dual nanoparticles be used for? So in the examples that you saw, gold nanoparticles have been used for DNA detection. What else can it be used for? So somewhere in the US, in the university, Rice University, there is this female scientist. Her name is Professor Naomi Hallis. And she came up with gold nanoparticles that could be put into the body and then be used to kill cancer. Okay? You can be used to kill tumors, destroy tumors. And the way it does it is, when you shine light onto gold nanoparticles, they give off a lot of heat. Okay? They give off a lot of heat, and so much heat that it can kill all the tumor cells around you. Okay, so that's actually already FDA approved, and there's a company. Okay, so I don't know when, but we might see that kind of therapy in Singapore sometime. I'm not sure how long it will take for it to come here, but it does have advantages over chemotherapy. Have you all heard of chemotherapy? It's some poisonous chemicals they put in the body, and it starts destroying a lot of tumors when activated. But unfortunately, chemotherapy is so destructive it can kill all your cells, good cells, bad cells, healthy cells. You don't want the healthy cells to be killed. So the gold nanoparticles are more targeted, and I won't go into the details of that, but there is this hope that they might be better than chemotherapy for some tumors at least. So the second part of the question is, what are the advantages and disadvantages of using gold nanoparticles? So one obvious, the advantage, I think you can already see in the application, that they can be used for DNA detection because they have a large absorption cross-section, and then they take in light really well. That same property was also used to allow them to be cancer therapeutic agents. Disadvantage of a gold nanoparticle is, for DNA detection at least, it turns out that using this nanoparticle is not the most sensitive. Okay? In the example that, I'm not sure if all of you took part in this competition, but the example that we had, it could only detect picomolar. Okay? Picomolar, what does that mean? In 1 liter, you will only have picomoles of DNA that you can detect. Now, if you use some other kinds of nanoparticles, for example, nanoparticles that give you fluorescence, okay, such as semiconductor nanoparticles, then your sensitivity can go down so low, it starts to be attomolar. Okay, picomolar is 10 to the minus 12, attomolar is 10 to the minus 18, so 1 million times more sensitive than the DNA, than gold nanoparticles, so it can be more sensitive. So that could be seen as a disadvantage. The other disadvantage may be that gold, to some people at least, might be too expensive. Okay, so if you can replace it with a cheaper nanoparticle, why not? Okay, so that's, I hope that answers your question. And I'll pass the mic over to my colleague to answer the other part. Yes.
You say cheaper nanoparticle, right? Then what other particle can be used?
Yeah, so For example, if you want to do the same UV-vis experiment to replace gold, you can actually use silver. Okay, not so much cheaper, but for some people, gold, well, I think at least it might be agreed upon internationally that gold is more expensive than silver. But you can actually use silver nanoparticles as well in the UV-vis experiment. However, gold nanoparticles are useful because you can actually see the color with your eye. If you use silver nanoparticles, you won't so easily be able to see those colors with your eye. But a UV-vis machine can actually still be used because the machine can detect wavelengths that our eyes cannot see. Okay, so silver is one example that's also used. We're hard-pressed to think of other metal nanoparticles that can be used at the moment. But so I shall leave that as that, silver nanoparticles. Okay, so any other questions? Okay, so I'll just pass the to my colleague.
Okay, we have a question here. The question is, why don't DNA on the glow particles and the glass surface directly bind together? I'm assuming that the question is, why won't the DNA on the glow particles bind to the glass surface? Now, the short answer to the question is yes. Okay, glass is made of what? Silicon oxide, right? So silicon do bind. It's a hydrophilic surface. It does bind DNA to hydrogen bonding. So this is actually this is more of a problem. Okay, so if you have a glass, how do you prevent the DNA from sticking on it if you want to develop a sensor? And there are ways to mitigate this problem through this problem is called non-specific binding. Okay, and you can mitigate this problem by coating the glass with a protein molecule or something. So you block off all the reactive sites. So when the DNA will only bind to the DNA that is coated on the surface and not on the glass surface. Okay? So the short answer is it does happen, but it can be overcome through engineering, okay? Through this form of non-specific, what we call blocking. Okay? You block the reactive site by using something that doesn't interact with the sensor properly. Okay? I think that's about it. We've come to the end of the British feedback. Thank you very much.
Thank you, judges. We will now give out the honorary mention awards where non-finalist teams with the best answers to different disciplines in the competition are recognized for their efforts. 2 honorary mentions will be awarded per discipline. For questions which involve more than one discipline, for example, in part 3, 3 honorary mentions will be awarded. Teams, please send a representative down to receive the award. honorary mention for chemistry goes to Team 2 from River Valley High School. The first honorary mention for biology goes to Team 2 from Westwood Secondary School. And the second honorary mention for biology goes to Team 1 from Victoria School. Before we announce the results of the finals, we would like to acknowledge the participation of all the teams who are putting out hard work in tackling the very challenging problems. In their own right, they are also winners in not giving up and in persevering to go. All schools please send a representative on stage to receive the certificates when your school name is being read. The schools who are with us today are NUS High School of Mathematics and Science. Please come down with me. Chongqing High School name. High Catholic School. Warsaw Institution. Methodist Girls School. Westwood Secondary School. Woodland Secondary School. from NUS High School.
The team comprises of Aditi Sauraji, Bel Somyar, Huang Fei Yang, and Felicia Tai Fei. And now for the moment we've all been waiting for. The 3 commendation awards, not in order of merit, goes to team from Woodland Secondary School. Comprising of Erika Danielle Team Funding Award, Le Sharafara, Aaron Go Ziyuan Flo, and Zhou Zihan. The team from Hwa Institution, comprising of Go Hongwei, Go Guo, Go Yangwei, and Go Yongwei. The last team from Westwood Secondary School, comprising